Along with some other people that I'll mention, in particular Ted Gilberts and Rick Adams. And a couple of, so what is the point of today? The point of today is, one, I want you to overall understand the evidence that supports why myself and other people in the field think that fats are a real taste sensation. It should be a sixth taste category, that there should be a sixth taste category for fats, for fatty acids. So that's the first thing, and I'll have slides. It's like, what do I need to know? This is what you need to know, what I just talked about, okay? So pay attention to those slides, make sure you write down those slide numbers, okay? The second thing is I want you to, I want to peel back the curtain a little bit and show you how this science is actually done. Like, how do we know all this stuff in this textbook that you're reading, right? And a lot of how science gets done is actually failure. We fail probably more times than we succeed. But you never hear about the failures because nobody talks about that, right? They don't get published. They don't get presented at conferences. Well, sometimes at our end of the semester research colloquium, you see some failures, but they don't really get talked about. And actually, Stuart Firestein is an olfactory researcher. He's written 2 books that are wonderful books. He also has a great TED Talk. Maybe I'll post the TED Talk on the homepage about failure and why science is so successful and how we learn probably more when the results don't turn out the way that we thought they would than when they turn out the way we thought they would, right? He also has a great book about ignorance, which is talking about the fact that we know less about science than we think we know, right? And that there are still many, many questions left to be answered. Every time we answer a question, it opens the door for more questions. So it's like we're never solving the problem, we're just slowly, incrementally learning more about it, right? And so, anyway, I want to talk about how we come to know this kind of stuff. All right, so, way back in the dark ages, when Dr. Pittman was graduating graduate school, I got a job here at Wofford, and when you leave, so when you're getting your PhD, right, you're working under a mentor, you're working under a PI, a principal investigator, on their research, their grants, their research line. And when you leave, you have to develop your own research. You don't just keep doing their research for them, right? And that's a big transition. And so as I was thinking about, well, what is it I'm going to do at this new place at Wofford College, and what sorts of research questions could we pursue, that sort of thing. I'm going to grab an A and a B cup and a spoon. I was very interested in the relationship. I spent my PhD looking at taste signals from the mouth and how that affects feeding, particularly with salt and with and in development. So we were giving rat mothers while they were pregnant low salt diets or high salt diets and seeing if that affected their offspring's preferences for salt and their taste responses to salt. Does salt during pregnancy, salt levels influence the development of the taste system? But I wasn't really interested in doing that. I was interested in how these signals sort of drive feeding behavior. And at the time, we were in the midst of an obesity epidemic. Not that we're anymore, but that was sort of the hot topic at the time. And so I was like, okay, well, what drives obesity? And there's 3 major things that drive obesity. Can you think of what they are? Reward of sweet food?
Taste sensitivity.
Taste sensitivity, so certainly. There's genes, right? So some people's genes make them more predisposed for gaining weight than other people's genes, unfortunately. Some people have, you know. But that's about 5% of obesity. Can you account for that? The other 2 things that are obesity is calories in, calories out, right? It's a pretty simple equation, right? Our body's really good at when we expend calories, we have to consume calories. If we consume more calories than we expend, our body stores those as fat. Really good at it because for thousands of years we couldn't get enough calories, really, right? We were very thrifty, right? We didn't have access. There weren't McDonald's and there weren't grocery stores and fast food places everywhere where we could just go get unlimited calories, right? So our body is programmed to take any excess calories and store them for us, right? And this overconsumption of high-fat food and high-caloric food was a main driver of where we were getting all these calories. So the question is, why do people want to eat that? And Aaron said, well, it's rewarding, right? Okay, it's rewarding. How is it? Is it just the nucleus of accumbens, just the reward system? Or is there something else, right? And so I had the simple sort of question. We know that eating fatty foods makes us obese. Why do we keep doing it? I'm asking you, why do we keep doing it?
It tastes good.
Because it tastes good, right? Fatty foods taste good. But wait, that's not a taste. But if you ask anybody why are you choosing to eat that, they say, because it tastes really good, right? So, you know, McDonald's French fries taste good, right? But they're soaked in fat. They contain a lot of fat. So we got into this question about, okay, is fat a taste? Could it be a taste, right? So the number one answer is palatability, right? That high caloric foods are our favorite foods to eat because they taste the best. All right, so question, does fat have a taste? So in front of you have two whipped creams, right? One is fat-free, one is not fat-free. I want you to taste each one and scan that QR code and answer A or B as to which one is the fat-free. A quick poll here. All right, so get your phones out. Hopefully that goes to a Google form. All right, I'm getting some responses. What was your answer? I'll flip flop it. All right, still going? I gotta refresh my screen. All right, has everybody answered that wants to answer? All right. So you guys are choosing which is the fat-free whipped cream, right? All right, so vast majority of you said that A is the fat-free whipped cream, and you were correct, right? Some of you said B were wrong, but why can you tell that? Why can you tell that? Right? Yeah.
Well, I felt like I could tell just by looking at them, because A was holding up its structure, like I could tell when we would like put it in the cup, it was kind of holding, and then B was a little bit more like melted and like smooth and like the texture, and that's what honestly kind of made me like the, oh, that is the best.
Okay.
I just thought you tasted better.
Yeah, yeah. So forever, fat has been relegated to what Mary Margaret said, which is a taste, which is a texture sort of perception, right? That it has, and it does have a textural perception, right? It does stimulate this filiform papilla on our tongue, right? And it does, but there are other things that can produce texture, like xanthan gum and other additives, right, that we can put in to mimic that sort of texture, right? If you ask chefs, like, about how they use fat, I don't know if any of you have ever worked in a kitchen, but the amount of butter that they put on food in the kitchen is ridiculous. There's like usually a huge vat of melted butter that they'll just throw it on everything. Why are they doing that? because that fat enhances the flavor of almost everything coming out of the kitchen, right? So if I were to give you a plate of sucrose and some Crisco, I talked about this, I think, back when we were first talking about taste transduction, right? Not appealing. Nobody wants to eat that, right? But if we mix them together, we get frosting. And if I give you that, you'd be like, yeah, I can eat some of that, right? Straight out of the can, okay? So apparently, maybe fat by itself is not so appealing, but when we mix it with other tastings, like sweet, all of a sudden it becomes this new, more palatable thing that we really like, right? So there was sort of this idea circulating amongst some of us taste researchers that there might be sensations from the mouth to the brain related to the taste of fat, right? Common experience sort of suggested that fat should have a taste. Okay? So if that's our hypothesis, right? The first thing, like when we talked about taste and smell, how did I group them together? What did I call it? What kind of senses? Chemical senses. So the first thing we have to understand is that fat is being transduced on the tongue. What are the chemicals that are in fat that are stimulating taste receptor cells? That's like the very first thing we have to understand. And it turns out that fats are made of these things called triglycerides. Lipids are made of triglycerides. The triglycerides are called that because they have a glycerol backbone and they have 3 fatty acid molecules attached to it. Right? This is the chemical composition of fat. And in our saliva, and in rat saliva, we have an enzyme called lipid lipase that can break the bond in between those fatty acids and the glycerol, so those fatty acids become disassociated and able to interact with things in our mouth. Whoops, there we go. And so Kawai and Fushiki, 2 Japanese researchers, back in 2003, did a study to examine, does lipid lipase have the ability to generate significant amounts of free fatty acids? So this whole, I should back up and say, back in the dark ages, Dr. Pittman graduated with his PhD and started here, it was in 2001. And so, so we were really at the forefront of when this was happening, right? There were people talking about this, but nobody had really done any research to show any evidence of it. And so this is a major breakthrough. And so what they did is they took triolene, which is a fat, that has a triglyceride, has 3 oleic acid, which is a type of fatty acid, 3 oleic acid molecules attached to the glycerol backbone. That's why it's called triolene. Okay? And they made it radioactive so that they could measure the disassociation of those bonds. And then they put it on rats' tongues for 1 second, for 5 seconds, or 10 seconds, and then they could measure the detachment of oleic acid from the triolene. And it turned out that within 1 second, they could generate about 53 millimolar, that's the concentration of oleic acid. So that number will be important in a few slides, right? So you can think about that. So really rapidly, put fat in your mouth, within 1 second, we generate a concentration of about 53 millimolar oleic acid. It goes up even more if you chew your food, right, to 5 seconds or 10 seconds, it goes up more. So now, we've shown that, hey, these free fatty acids could be the chemicals that are being detected. If we're looking for a target chemical, these look like good target chemicals. All right, so corn oil is sort of the prototypical dietary fat. There we go. And so our question was, okay, if we want to make this relevant to feeding, what are the fatty acids that are found in corn oil, right? What is the composition? And it turns out that there's mixtures of these triglycerides, and it's what makes corn oil different from olive oil, or different from sesame seed oil. They have different combinations of these fatty acids. So it turns out that corn oil has two predominant fatty acids. Oleic, that I mentioned on that last slide, about 31% of the fatty acids in corn oil is oleic. 52% is linoleic. So these linoleic acid and oleic acid make up over 80% of the free fatty acids in corn oil. So those seem like pretty good targets, right? One of the things we were interested in was isolating the chemical that we thought was being transduced and only presenting that. We didn't want to give rats corn oil because it has texture, it has nutritive qualities, it has all this other stuff associated with it. We wanted to see if we just took the chemicals that we thought were being transduced, could we do research with those? So these seem like two good target chemicals. Olive oil has predominantly oleic acid and much less linoleic acid. It's one of the differences. So if you were to taste olive oil and taste corn oil, they taste fundamentally different. It's because of that change in the percentage of fatty acids. Yes, Christine?
What is corn oil like commercially? Is that like canola oil?
Yeah.
Okay.
Okay, so around the same time, so in 1997, so a little bit, this was when I was still in graduate school, one of my colleagues in the taste community, Tim Gilbertson, was doing some really interesting, what I thought was interesting research, where he was extracting taste receptor cells out of rats' tongues, putting them in a dish, and doing patch clamp recordings from those. So essentially what he was able to do is stick an electrode into this taste cell and then put linoleic acid in the container with it and measure if there was depolarization of that taste receptor cell. Could the presence of this chemical be transduced and cause depolarization in the taste receptor cells? And what he found is that when he did that, not only could he measure depolarization, but he could tell that it was a potassium channel that was being blocked by the presence of linoleic acid. And potassium, again, normally flows out of the taste cell. And so by blocking this potassium channel, it caused depolarization and could cause neurotransmitters to be released. So he was starting to hone in on a possible transduction mechanism for linoleic acid. So these are, he's putting linoleic acid on, and he's seeing the relative outward potassium current goes down. That means it's getting actually more positive in the cell because we're shutting off that current, we're turning off that current, right? So this is arachidonic acid. Oleic acid surprisingly didn't do that. Linoleic acid did, palmitic acid did not. So he found that some free fatty acids could activate this receptor, but other fatty acids didn't. It's kind of interesting. Also, when he would inject current into the stimulus and then add linoleic acid, it increased the depolarization, right? And so this led to the idea that sort of the theory of why does putting fat Crisco with sucrose enhance the taste of sucrose? Maybe because if this is sweet and then we add fat to it, it becomes even stronger. It becomes even more potent. There's more depolarization. Right? So it could be a mechanism for how fats enhance salt and enhance sweet taste. All right, so this is Tim. He's a big Denver Broncos fan. I won't hold that against him too much. But he said, all right, where in the body, is there any research or evidence in our physiology that free fatty acids can be detected? And it turns out that in some non-taste tissue, like the immune system, the heart, the brain, the pancreas, the intestine, there are these metabotropic receptors that can bind fatty acids. And he's like, well, I wonder if any of these things are actually found in the tongue, too. No one's ever looked at this, right? It's a whole brand new research question that we were looking at. And so he was able to again, do some experiments to identify roles of these different, or the presence of these different G protein coupled receptors or metabotropic receptors on lingual tissue. So this is it's showing expression in fungiform, folate, and circumvallate papilla. We can see that there's amounts of these metabotropic receptors found in these different things. And so he then now has identified a second way in which potentially fatty acids could be detected or transduced on the tongue. One, they can block this delayed rectifying potassium channel. Two, there were metabotropic receptors also present in the taste tissue that could be activated and cause calcium to be released and cause neurotransmitter release. All right, so we came up with this model, and it turns out these are the potassium channels. They're found everywhere on the tongue. But these G protein coupled receptors, some of them were found more on the anterior, some more on the posterior. This CD36 is another mechanism. So you came up with this whole model of multiple ways in which these fatty acid chemicals could be transduced on the tongue and cause depolarization and might result in taste signals being generated. Pretty cool stuff. All right, my first slide that you need to know. So write down 11, right? What do you need to know about this, right? You don't need to know about patch clamps and the G protein coupled receptors and the names of all this. But what you need to know is that fatty acids have a way, right, to depolarize taste receptors. They can do it, one, by inhibiting potassium. They can do it by increasing calcium through G protein coupled receptors or metabotropic receptors. That GPCR is the same thing as saying metabotropic. And that CD36 is actually a fatty acid transporter protein that may help recruit these molecules to the taste receptor cells. Remember I said that there were, in the olfactory mucosa, there were molecules that could kind of grab odorants and pull them in towards the cilia? It looks like that's what this role of CD36 is, right? So, a helper mechanism and two mechanisms for transduction, metabotropic and potassium channels, right? And that they're differentially expressed on the tongue, right? So that was sort of this paper and this series of papers that came out in the late 1990s, 97 to 2000, sort of were the basis for when I came to Whopper. I was like, well, hey, I know how to do psychophysics in rats with taste. We could devise some behavioral experiments to see if behaviorally animals show evidence of what Tim's looking at in these isolated taste bud receptor cells, right? Some of the criticism, one of the things about science is that it's very resistant to change, right? It's very hard to get new ideas accepted. You really have to, and that's a good thing because we don't want the scientific knowledge base just whimsically changing based on one study that came out, right? You really have to have a preponderance of evidence for people to really accept change, right? So a lot of criticism of Tim's work was, yeah, that you're taking these, this isn't really how it works in the tongue, you're taking these taste receptor cells out, you're isolating them in this dish, petri dish, and you're putting these chemicals on it. And yeah, you can see these things, but we don't really believe that happens in an intact taste receptor in the tongue, right? So the questions that we wanted to know is, overall, right, does our taste system detect fat in foods and motivate us to continue consuming those through palatability, through saying that that's a positive thing, right? And so our questions were, are fatty acids detectable? Do fatty acids affect the ingestion of other tastings, right? So when we put fat with sugar, does it make that sugar more palatable? Right? And those were our first two questions. We're gonna ask some more questions in a few minutes. So we started out actually with this second one. And again, we didn't want to take Crisco and mix it with sweet, right? We wanted to take isolated chemicals, fatty acid chemicals, add just linoleic acid to tastings, and see does it change the way rats consume those tastings. All right, so we get to our conditioned taste aversion paradigm. So here's, I know it's hard to imagine young Dr. Pittman with gray hair coming to Walter, all excited, like, what can I do at this new place? I have no equipment, no money. Oh, well, conditioned taste aversions are pretty easy. All you have to do is give rats something to drink and then inject them with either a saline injection, in which case they'll drink that stuff the next day, or we can give them lithium chloride that makes them sick, and the next day they're like, no, I don't want to drink that anymore. But they can only do this if, one, they get sick, and two, they can taste the thing that you're giving them. If they can't taste it, right, then they won't avoid it, right? All right, so here we are giving rats 2 different samples. We're measuring their licking, right? And so our very first experiment was really pretty simple. It was just a 2-bottle preference test. I didn't even have this fancy equipment to measure licking. We just measured the bottle's weight, right? How much do they consume in 24 hours? Really, really simple experiment. But this was the first time anyone ever showed the ability of rats to detect and avoid here, right, fatty acids at very small quantities. I forgot which order I was doing. I'm actually doing the first bullet point there, which is, can rats detect fatty acids? We'll talk about adding them to tastings in a second. So over here we have linoleic acid, and we have different concentrations, and we wanted to see if we could define a threshold, psychophysically. And what we did was we found that if we gave them 44 micromolar, which is a really small quantity of linoleic acid, they treated it no differently than the control animals. They consumed it. If we gave them a taste aversion with 66 or 88, they started to avoid it, and they avoided the 88 more than the 66. So we were able to identify a concentration at which they acted like they couldn't taste it, and a concentration above which that it looked like they could detect it and they avoided it. And then we did the same thing with oleic acid and found similar things. Now, who remembers what concentration Kawai and Kushiki said got generated within 1 second putting trialine on the tongue? 53 what? Millimolar. 53 millimolar. That is 100 times more than this, than 66 micromolar, right? So we're essentially saying plenty of fatty acids can get generated almost immediately on a rat's tongue to allow them to detect and avoid it. Even though we weren't doing that, we were using these concentrations that were well below what could get generated on the tongue. Yeah?
I know during the smelling, we talked about how rats are better at smelling because they have more brain areas. Like, how much of this rat research can translate to humans if they have
that's a great
more
question.
brain stuff?
Yeah, that's a great question. And that was one of the critiques that we got about our research was, okay, well, rats can do this, but people can't, right? Was the question. Unfortunately, people don't like you to do conditioned taste aversions with them. They kind of not a great thing, right? I mean, I would have done it if some people would have volunteered, but it's, no. So that was a critique, right? And one of the big critiques was that humans don't have the same lingual lipase in their saliva that rats do. It wasn't so much a brain thing as it was they didn't think that people generated the same amount of fatty acids. So that's a question we're going to get to. Great. All right. So cool. We were the first lab, little old Walter College, first lab in the world to demonstrate that rats could detect and avoid micromolar quantities of the chemicals in dietary fat, right? Suggesting that they taste those things. Pretty cool. So yeah, this is me before I had kids and gray hair. Some of my first students back in the early 2000s, we presented this research at our annual research conference that I'll be going to actually next week, no, 2 weeks from now. And we published this, right? back in, we did this in the early 2000s, we published it in 2006. And by that time, I had gotten some grant money, I was able to buy some fancy equipment, and so we have this thing that we call the MS-160 or the Davis rig, and we still use this today in the lab, right? We put these rats in here, and the shutter will open, we can put different bottles in front of them, and when the shutter opens, the rat will start licking. Lick, rat, lick. All right, he's licking now. Computer's counting the number of licks. Rats lick really fast. They lick about 7 times a second. They really hit that, right? Computer counts that, and so we can measure how much they lick in 16 different taste solutions. Very short trials, 15, 30-second trials, so that we know they're not getting feedback from their gut. And these fatty acids don't have a lot of nutritional quality because, or calories, because we're using these micromolar quantities. Right? So our next question was, okay, they can form taste aversions, it looks like they can detect it. What about Tim's theory that fatty acids enhance the taste of other tastings? Right? So we tested them with sucrose and with glucose. This is sweet alone. If we make something more sweet, rats lick it more. Then we added linoleic acid, and when we added linoleic acid to it, it enhanced their licking. They licked it like it was sweeter, like it tasted better with the presence of 88 micromolar, very, very small quantities of these isolated free fatty acids. Right. We did the same thing with oleic acid and saw a similar increase, but it was much smaller. It was much smaller. And when we did the original taste aversion studies, they seemed like they had about the same threshold. But as we started doing more of these studies, it looked like linoleic acid was maybe a stronger activator of this system than oleic acid was. We also did it with things they don't like. So we made them thirsty and gave them salt, and they would drink salt at low concentrations, but if we make it salty enough, they're like, yeah, I don't like that. Even though I'm thirsty, I'm not going to drink that. Same thing with sour, same thing with bitter. And when we added linoleic acid to it, it looked like it enhanced the taste. It made things that they normally would consume stronger, and they started to avoid it earlier. They had a stronger aversion to it. So both of these things look like fatty acids increase the perceived intensity of other tastings, which makes sense if you think about that idea, that theory of why does adding fat to sucrose to create frosting make it more palatable. All right, so this is the group that did that. We published this back in, when was that? That was in 2006 too. So we were doing both of these about the same time. We actually did that later, but it takes a while sometimes to get things published out there. All right, so next important slide, 23, right? What do I need to know? There's evidence, right, that linoleic and oleic acid increase licking to appetitive stimuli and decrease licking to aversive stimuli, which we interpreted as these small micromolar quantities of chemicals found in dietary fat enhance the perceived intensity of taste. Also, linoleic acid produced greater effects than oleic acid. All right, good. All right, so we can check that off. We can check off the taste aversion stuff, right? But as I mentioned that, those initial taste aversion studies were pretty basic, right? And part of the reason it took so long to get those published is we had to convince reviewers that it was actually taste signals that were driving this, and that they weren't using what we call post-ingestive feedback from the gut, right? Because we gave them 2 bottles of 88 micromolar linoleic acid, one without, one with, right? And they had 24 hours to drink that. So they had plenty of time for maybe receptors in the gut that we know exist for fatty acids to recognize them and stimulate some reward signal to the brain. And maybe that's what caused them to be able to detect it and not taste. That was one of the big criticisms. We were able to get it published in, But I said, okay, if they're going to say that, I know that I can go in and I can cut the chordotympany nerve because it runs right underneath the eardrum, right? So I can go into the rat's ear canal and eliminate the chordotympany nerve. I can go in and cut it, and you will no longer have signals from the anterior 2/3 of the tongue, right? And if I do that, and there's a taste signal in that nerve that rats are using to detect and avoid fatty acids in a conditioned taste aversion, we should impair that, right? We should compromise their ability to do that if it's taste and not something from the gut. So that was the next experiment that we came up with. And we also wanted to use really short trials, again, so that instead of 24-hour tests, we're now using 8-second lick tests in that lickometer, right? And what we found is we could show a taste aversion to linoleic acid. Again, they avoided it at 88 and 176 micromolar. They didn't avoid it at 44, right? Over here, this is oleic acid. What happened here? Did they avoid it at all? No, they didn't avoid it at all. They didn't show a taste aversion at all. And we were like, oh, that's maybe problematic. It's like, well, why are we, and this is one of our first failures, right? So you learn more from failures, maybe, than you learn from success stories, right? And so we're like, okay, well, why might they not be able to do this? Well, we already have some evidence, maybe, that oleic acid was a weaker stimulus than linoleic acid. But if that's the case, maybe 8 seconds isn't long enough if it's a weaker stimulus for them to detect it. What happens if we give them 30 seconds? And so we gave them 30 seconds, and look what happens with linoleic acid. Now they can detect and avoid it at 44 micromolar, right? So again, just like you saw with the thermal threshold lab, which I'm working on grading very diligently, it's probably gonna be next week before I get this back to you. I'm working on it. The method, your methodology can affect your ability to measure thresholds. So now when we make it 30 seconds, rats have long enough, 8 seconds, they started licking the 44, and by the time the 8 seconds were up, they hadn't stopped licking. But if we gave them 30 seconds, they licked it for a little bit, and then they're like, yeah, that's the thing I got sick to, I'm gonna stop licking that, and they avoided it. Right? What happened with oleic acid? No effect again, right? We're like, wow, okay, maybe that's not it. So what are the two things that have to happen in order for someone to demonstrate, an animal to demonstrate conditioned taste aversion? Time. What are the two things that has to happen?
They have to taste it, they have to recognize it.
They have to taste it, and they have to have gotten sick to it. If you don't have those two things, you can't show a taste aversion. This is looking like they didn't taste it, right? But what if it's such a weak stimulus that they're not associating being sick with the oleic acid, right? And they're able to associate it with linoleic, but not with oleic acid, right? Oh, I'll get to that in a minute. But remember, we cut the nerve, and when we cut the nerve, this is what happened with linoleic acid. We couldn't see it with oleic acid because they weren't showing us any aversion. But when we cut that nerve, they stopped avoiding it, or they avoided it at lower levels because they still had that posterior part of the tongue intact, right? But we were able to show, hey, compromise the taste system, compromise their ability to detect this stuff. So now we really are providing pretty solid evidence that there's a signal in the taste system that animals are using to detect these fatty acids. All right. They were still able to avoid, at some concentrations, again, they have the posterior part of the tongue intact. All right, where are we going here? Here we go. All right. So our next thought, I was really troubled by this oleic acid failure. I'm like, what's going on here? This shouldn't be happening, right? And so we thought the only other thing that could be happening is maybe because it's so weak, they're not associating getting sick with oleic acid. So even if they're able to taste it, they didn't form that association. So before, we were just doing a single injection, a single pairing of lithium chloride with that. We said, okay, we're really going to make sure that they form this association. So we did 3 consecutive days of giving them linoleic or oleic acid and making them sick. And then the next day doing it again, and the next day doing it again. So they really wanted to see if that test that theory. And when we did that with linoleic acid, we generated much stronger taste aversions, and we went lower in our concentrations because we showed they could detect it at 44. And we were able to show a threshold of around 5 micromolar, between 5 and 20, with 3 pairings of conditioned taste aversions, right? Now when we did that with oleic acid, we're able to show that. So it did appear that oleic acid is a weaker stimulus, and it took more than just a single conditioned taste aversion pairing in order for them to associate that weak taste with getting sick. Right? And again, you can see the difference. Their threshold's much higher, around in between 20 and 50, where linoleic is much lower at between 5 and 20. Stronger stimulus, weaker stimulus. When we did the first experiment cutting the nerves, we cut the nerves and then did the experiment. And so people were like, well, you compromised the taste system before you did it, so that's why they didn't show the taste aversion. So here, we did this strong taste aversion, and then we cut the nerve afterwards. So we were able to show, hey, in this group of rats that are avoiding linoleic and oleic acid, now we're going to take half of them and cut their nerve and see what happens. And when we did that, the half we didn't cut their nerve still show an aversion several days later. The half we cut the nerve, it eliminates that aversion, right? So that was one of the challenges of this. We had to form a really strong aversion because we had to test them, show they avoided it, that same day cut the nerve, let them recover a couple of days and retest them, and hope that that aversion was still there. And so these are the control animals that just waited 4 days and then got retested. These are the animals that had their nerve cut and 4 days later we retested them. So now we're really showing people, it's like really, what I thought was pretty indisputable evidence that people couldn't argue about, that there are signals in the cortex empty nerve that allow animals to detect fatty acids. All right, so we published this again, presented it at research with several students that went down there with me. I forget what this is published on. There's a date. Well, it was accepted in 2007. So all this is happening in like the mid to late 2000s. All right, so slide 33. Okay, it's the third slide you got to pay attention to, right? We showed that micromolar, very small quantities of linoleic and oleic acid could be avoided in short trials after a taste aversion. If we increased the trial duration, we were able to show lower thresholds. If we increased how sick they got, we were able to show lower thresholds. So all of this is following lock and step with what we would predict with psychophysics and taste. Eliminating the cortex tympani nerve, eliminating the signal from the anterior 2/3 of the tongue, impaired their ability to do this, right? And so all of this, we think, suggested that yes, indeed, the chemicals that break apart when you consume lipids, fats, free fatty acids, can be detected at very small quantities through the taste system in rats, right? And so that's the big thing. All right, we also showed that it looked like linoleic acid was more salient, right? Produced stronger, more maybe important signals than oleic acid. All right. So, check that one off, right? And this took us about 6 years, produced 3 publications, and at the same time, Tim is still working in his lab doing these transduction mechanism experiments. And he started looking at genetic influences on taste sensitivity. And so, here's Tim. He was measuring taste receptor cells from a strain of rats that doesn't get obese, a lean species of rat, strain of rats. and a strain of rats called the OM strain that get obese. And it's like, well, why do these rats get obese and these rats don't? And it turns out that it is a preference in the type of food they like. So if we give them a high protein diet, they don't show a difference. If we give them a high carbohydrate diet, right, the lean rats prefer that. But if we give them a high fat diet, the obese rats really prefer that. So it looked like there was a difference in fat preference between the obese rats and the lean rats. And so Tim's publishing some of this and showing differences in the sensitivity of taste receptor cells from these two strains of rats. So I contacted Tim and I'm like, hey, any chance you could send me some of those rats? And he was like, sure. I was like, I'd like to do some behavioral tests. Let's look at behaviorally if we see these same things. So he sent me the rats. I said, thank you. He said, you're welcome. We have this collaboration together, right? So our next question was, if we look at these fat preferring rats and these lean rats, non-fat preferring rats, do they show different sensitivities to fatty acids through this taste aversion paradigm? And so we did the test. We made them sick, gave them fatty acids, and we were also interested in males and females because there was some other research that suggested that females were more sensitive to the taste of fat than males were. And it turns out that there was no difference in the sexes or the strains, right, in their ability to detect and avoid it. And I was like, man, that stinks. That's not what we predicted. It's kind of failure again, right? But I was like, they're showing a really strong aversion here. And if we continue to give an animal something that they're sick to, right, eventually, without making them sick again, right, we can cause extinction of that condition, that learned aversion. They sort of unlearn that makes them sick. They learn it doesn't make them sick. So any of you that have a taste aversion to something, if you want to consume that again, you just have to power through and keep consuming the thing that you don't like without getting sick. And eventually you can eliminate that taste aversion. So I said, well, what if we look at this as it gets weaker? Let's continue testing. Let's test them on day 2, day 3, that sort of thing. Yes, extinction. So on day 2, we actually start to see sex differences emerge, where the female rats are showing stronger continued aversion, less extinction, than the male rats are. And we're starting to show some strain differences where the OM rats are showing more aversion in both strains than the lean rats. And by day 3, we were extinguishing most of that aversion, so they're starting to come back up to normal levels, right? So it turned out that this day 2 was kind of this sweet spot where we could actually show that there were subtle differences. And conditioned taste aversion is a very strong learned condition. So here we weren't able to see these subtle differences, right? They were just like, yeah, that thing made me sick, I'm not drinking it. But on the second day when they're like, yeah, that thing didn't make me sick yesterday, maybe it's not so bad, we're able to see some differences in sensitivity, right, to being able to detect these fatty acids. So here's the day 2 data. If we look at just the animals that got sick, and what we found was within the strains, the females always showed stronger aversions than the males. And within sexes, the obese rats always showed more sensitivity than the lean rats did, which is pretty cool. All right. We then said, well, what about exposure? Does exposure to dietary fat influence this? And what we found is that if we expose, so the data you just saw was on regular chow. If we gave them high-fat food for a period of time, right, the lean rats were able to now show as strong an aversion as the OM rats. So how much? So genes matter, but also how much fat you're consuming primes the taste system, right, and can allow you to be more sensitive to that, right? So raising these rats on a high-fat diet or a regular diet influenced them, made them more sensitive to fat than the genetically obese rats were, actually. All right, so we got 2 publications out of that with Tim as a co-author on both of those. All right, so again, slide 43. Pay attention to the slides that have the summary points on it. I don't really care if you understand the data, how we got there. I mean, I'd love for you to understand that. But what I really want is for you to get the take-home points, right? Which is obese-prone rats show stronger avoidance than obese-resistant rats. So genes do influence our taste perceptions of fats. Females show stronger avoidance than males, right? There are similar detection abilities, but these are subtle differences in sensitivity. Subtle differences in sensitivity to fatty acids. All right, so all of this is going on. We're now into like 2010. And people are like, okay, fine, we'll accept that rats can do this. Humans still can't. We don't believe that it's a human taste, right? And so Rick Mattis at Purdue studies human psychophysics, right? And I will point out that one of the biggest critics of this research and one of the people, strongest voices that humans couldn't detect fat was Linda Bartoshuk, the person that wrote your chapter. It's one of the reasons I think that they don't really talk about fat as a taste in the chapter, which I think is a real disservice that she's got that sort of influence over all the S&P classes that teach out of that textbook, because there's really pretty good evidence, as I'm showing you, from not just our lab, but other labs, that this is a real thing. So Rick Mattis does human psychophysics like Linda Bartoshuk. And he's giving linoleic acid to humans and asking them, can you taste this? And is able to define a threshold, a concentration threshold, for linoleic acid, for oleic acid, for all these different types of free fatty acids, right? And it turns out that humans are less sensitive than rats, but humans can still detect it at about 1 millimolar linoleic acid. So not at the micromolar, not 5 micromolar that we were able to show with rats, but humans can detect it about 1 millimolar, which is still a pretty small amount and probably well under, hopefully, the percentage that humans produce when they put fat in their mouth, right? He did some really exquisite things where he put the linoleic acid on people's tongues. He had them sniff it to see if they could detect it by smell. He wanted to isolate these different things. He even, like retronasal olfaction, he took like a really small whey boat and put the linoleic acid in the whey boat, and then put the whey boat on the back part of their tongue so that it could go through the retronasal passageway without touching the tongue. And was able to show that yes, we can detect it in multiple ways. It's not just taste, there is a role for smell in detecting fatty acids as well. Because again, it's really hard, especially with non-animals where we can do things like cut your taste nerve, right? With humans, to tease apart flavor from taste and smell. Right? But he was really good at doing that. All right, so he shows this, and then people are like, yeah, okay, fine, but humans don't have the same lingual lipase that rats do. We don't believe that they can break this down, right? We don't believe that these things get generated like they do in rats, right? So that's where, once again, our lab stepped up and said, well, can we do something similar to what Kawai Fushigi did? We can't obviously use radioactive trinylene on humans' tongues, but can we do something similar, right? And so what we did with these 2 students, and we presented this research, is we had people take corn oil in their mouth and swish it around and then spit it back out, and then we measured what they spat back out to see if there was an increase in fatty acids in what they spit out compared to what went in their mouth. And we were saying, okay, maybe we can measure through chemistry the production of these free fatty acids. And this was a collaboration with the folks up on the 3rd floor. They were helping me with some of the chemistry techniques of how do you measure how the quantity of fatty acids in someone's spit they just spit out of their mouth after swishing this corn oil around in their mouth, right? And so we did this at different times. So we had some students come in, don't you want to be part of this study, and swish corn oil in their mouth for 10 minutes or 20 minutes and then spit it out. We also had them do it for 30 seconds or 1 minute or 2 minutes or 5 minutes. And what we were able to do is show that the concentration of fatty acids increases the longer you swish it around in your mouth. But at 30 seconds, we're able to generate 2 millimolar concentration of fatty acids that we could detect, right? We did this with corn oil, we did this with olive oil, and we did it with mineral oil, which doesn't have any fatty acids in it. And guess what? When you swish mineral oil in your mouth and spit it out, you can't measure any fatty acids. So we thought this is pretty strong evidence, right? That at around 3.5, definitely under 4 millimolar, that we're able to produce these within a short amount of time in our mouth, right? Not the most sensitive of techniques. Probably, again, the type of method you use to measure this sort of stuff affects the threshold that you find. I think if we had, we were using acid-base titration in order to measure the production of these fatty acids. It becomes problematic to put a sample from someone's mouth that has spit and corn oil in it in a machine like a GPCR that's going to, or a mass spectrometer that's going to measure really small differences. Those machines don't like that sort of thing going through. And so I think if we had better techniques, we'd be able to show a lower threshold. But at any rate, we were able to demonstrate in the low millimolar concentrations, right, that this stuff was being produced and likely could be tasted by humans. All right. So this we actually never got to publish. We tried to publish it, and people didn't like that that acid-base titration technique, I guess, and we weren't able to publish it. So I don't think it's a failure. I think we showed some good evidence of that. Other people since then have shown similar things with humans being able to produce these fatty acids as well. All right, so review points. Be able to describe the transduction mechanisms, right? Be able to describe the 2 experimental models that show evidence to support that we could detect this. That was the CTAs and the increased in perceived intensity, right, when we add fatty acids to other taste notes, all right? Be able to describe that role, be able to describe the behavioral evidence supporting genetic and sex differences, right? That the first day of the conditioned taste aversion was so strong we couldn't see small, subtle sensitivity differences, but as we showed extinction, those differences in sensitivity emerged, right? And understand the evidence to support that humans can detect fatty acids and produce those in their mouth. All right, that's it. I felt like I went through that really quick, but it's okay. I get excited about this. All right, any questions? Cool. Have a good weekend. Don't forget to study some this weekend. We have an exam next Thursday. Have questions for me. Tuesday is your opportunity to ask questions in a review session. And then just throw away your samples that you leave.